Hello, this is Mickey Urdea for Halteres Associates. Welcome once again to another episode of Interesting Stories in the History of Diagnostics. Today I'd like to talk about spatial biology. This is a major trend in life science research right now, and I think it's going to have profound implications. Those of you who don't know about spatial biology, it's viewed as the study of cells within the context of their tissues, including their morphology and biochemical status. That, by the way, is my definition because I found like 10 different definitions, which were all very different from one another. So go with it, if you will. I'll cover where the scientific community is with our understanding of overall human biology at the biochemical level, because that's really pertinent here. And then I'm going to transition to how this is being used to understand our biology on the cellular level and then at higher levels as well, such as organ. The implications of the new depth of biological understanding that's unfolding right now with spatial biology is going to have a major and profound impact on the future of diagnostics, which I'll discuss later at the end. So let me talk a bit about where things stand with the understanding of human biology. And I think you're going to be a bit surprised, and I know I was when I started looking into this, at how little we actually know about a lot of this. So I'm going to talk about this on five levels. One is the genome, the transcriptome, the proteome, the metabolome, and then, I don't know, maybe we'll call it the cellome, and how that then is being brought together in a spatial biology context. So the human genome, we all think about it having been completed in 2002, but actually it wasn't completed until last year, 2023. That's the so-called T to T, telomere to telomere sequence. And, it's one composite genome in that particular case. It's kind of like having one sample of a new plant, an herbarium specimen, and that's the only one anybody's ever seen except the explorer. So, you have to build on that. So now, with improvements in the speed, breadth, and cost of NGS, we'll continue to sequence populations of people, then all individuals, and then variations within an individual over that person's lifetime, including mutations as they arise and mosaicism, which is where there are multiple genetic lines within one individual, which is more common than people realize. So, what are the implications of all this? What do these new variations mean on our health? How do these things change over time as we age? And what are the implications of that? That's going to be a very long set of experiments over a very long period of time. Look at this now at the transcriptome level, where we're looking at the RNAs, which are read from the genetic code. At the point in 2002, where the first major portion of the sequence was revealed, we came to the conclusion that there were 22,000 proteins that were coded for by specific mRNAs. That's about 5% of the 3 billion bases in the human genome. So not much. Today, we realize that probably at least 75% of the genome is transcribed. It's transcribed into direct mRNAs, often with introns, alternate splicing, non-coding RNAs, which are becoming more and more important in the area of cancer, small regulatory RNAs, etc. The more we look, the more we learn. So what do we need to learn? Well, what are all those mRNA variants doing? And what do we know about these non-coding RNAs overall? And we need to take into consideration also the fact that the non-coded DNA is important here. We're finding more and more that there are both activating and depressing proteins which bind to certain portions of the genome to stop expression. And we know that there are other applications in scaffolding of molecular structures. A lot of other things are going on that are not coded for directly by the classic gene. So I mentioned before that based on the Human Genome Project and work since, we believe that there are approximately 22,000 proteins which are expressed. In 2002, when we first knew that, we knew about what 60% of those proteins did. Today, we know about what 80% of them do, which means that we've still got something like 440 proteins that we don't know the function of. That's a lot more that needs to be understood. And I think one of the most amazing situations is the human metabolome, which has been cataloged first in 2007, believed to contain about 2,000 different metabolites that were entered into the catalog. But since then, in the 2022 catalog, we found that there are approximately 200,000 entries. But most recently, report last year, There may be as many as 1.5 million. So, what are the contributions of all these molecules? What are their functions and pathways? What do they do? And how are they related to disease state and wellness? There's so much more that we need to know. It's just astounding how much we don't know. So it's almost like having, you know, a 1,000-piece jigsaw puzzle, and we've now got about 100 pieces for it. There's a whole lot more that we need to understand before we can understand the full picture. Now let's take it up a level. Let me talk a little bit about cells as on our path to the overall notion of spatial biology. And of course, there are many ways in which those cells are going to function, but we first need to understand how many cell types are there. Since 2016, there's been a global coordinated effort towards creating a list of all the known cell types within the human body. This is referred to as the Human Cell Atlas Project. It's been coordinated very nicely because people sign up for a particular organ or cell type or tissue that they want to focus on, and then they start doing the appropriate work. This requires both imaging of cells and isolating and collecting cells of interest and characterizing those on a cell-by-cell basis, so single-cell analysis. I'll talk about that a little bit more in a moment. A major tool for this cell identification has been transcriptomics, really looking at the type and quantity of particular mRNAs within that particular cell. In 2010, one of the first reports of the number of cell transcriptomes was reported, and we had about 100 of them, 100 cells, 2010. 2015, we had about 100,000. And here now, this last year, in 2023, 39 million had been reported, with a total of about 300 different cell types. And the number of cells just keeps going up. We now have hundreds, and every time I turn around, somebody's identified another group of cells within a particular organ. The goal of the Human Cell Atlas project is to develop 1 billion single-cell transcriptomes. So that opens up a whole lot of new questions. What do each of these cell types actually do within their specific locations, within that specific tissue? Does that change the function of time? How do they function to maintain proper health? How are they altered by the environment? Our genetics before modification by the environment, our age, potentially diseases and treatment. And I think a key issue that people are struggling with right now is how do we actually define a cell type? A transcriptome might actually be transient within that particular cell type. We're really not identifying one cell type versus another in that way. So, it could be kind of like trying to determine someone's job by looking at what clothes they're wearing today. Today, they're wearing gym clothes. Tomorrow, they're wearing scrubs. What is the actual type of person that we're talking about? What is their job? What is the job of that cell? What's the ultimate way in which we want to define cell type? So, armed with the expanding knowledge of the human genome, the transcriptome, the metabolome, the proteome, the cell atlas, it's become possible to understand single cells in the context of their organization in tissues and ultimately organs. And then finally, the whole organism. We're starting to learn things from this understanding of cells within context that leading us to understand biology in a different way on a different level. For instance, pediatric and adult kidney cancers originate from previously little known cell populations within the kidney. There's been an atlas of the GI tract cells that's helped to permit us to understand small cell populations related to inflammatory bowel syndrome. There's now a human single cell atlas of the lung that has been used to identify novel epithelial cells, including asthma-related cell subpopulations within that. So I think, like genomics has done over the last 2 decades, the use of spatial biology tools is likely to expand dramatically over time from results from one composite human to populations and to specific individuals. There's a lot going on. Now I'd like to talk a bit about how the heck are we doing this kind of work and what's it going to lead to. But before I do that, I want to just point out one thing I'm leaving out that I'd like to come back to in another episode, which is the microbiome. Of course, we don't just have our normal somatic cells. We have all of these riders, if you will, within us that are truly part of us. I'm not addressing that here right now, but I'll come back to it because it is another perspective that needs to be added to the perspective we're getting by looking at somatic cells. So much of what's been done to date to look at the the function of a cell has been a reductionist approach. It is to take a tissue sample and to actually try to isolate particular groups of cells within it by dissociating them from the tissue, and then using those single cells to conduct whatever the type of analysis you might like to do. This typically requires a very specific type of sample prep, then isolation of individual cells, and most of the protocols nowadays involve some kind of a molecular barcoding for particularly mRNA and from the proteomic analysis, so that we have some way of knowing when we look at a large number of cells with one analysis, which particular cell that we put into that group they came from. So molecular barcoding is very popular for that. So in the case of looking for mRNA, most of that work is being done today with NGS, so-called RNA-seq. Very popular. And in this particular way, you're looking at having labeled the RNA from individual cells, combining all those mRNA populations back together and doing one large run on the sequencer, and then counting up the messages for the individual cells that were in the total population. In the case of single cell proteomics, this is a rapidly evolving field. And typically there are two different ways in which people are doing this. One is to use antibodies or other types of binders such as aptamers and actually look at the specific protein content from a cell. You can totally disrupt the cell, look at the proteins individually using these tools, and determine how much and which of each type is present. For this type of proteomic analysis, there are three major technologies which are used. One from the company SomaLogic, which is an array-based method using aptamers that have been designed to bind particular proteins. They can do more than 7,000 of those today, and that is on array. Then the companies Olink and Alamar that are using solution-based methods, usually with antibodies, and then using an NGS output to determine the amount of each particular protein that's present in the sample. And again, using molecular barcoding. The other method, which has become more and more advanced now and is being used much more extensively than I would have expected it to be, is actually mass spec. We've had mass spec for a long time. People have used it for looking at peptide breakdown products of proteins. The problem has been typically the number of peptides one has to look at, and secondly, then the sensitivity of those particular methods. You needed typically a pretty large amount of sample to detect that. Well, that's all changing now with the advances in a number of different mass spec systems from companies such as Bruker, Danaher, Waters, and then the laboratories which have adopted those particular mass spec systems and developed methods to improve sensitivity and specificity of the technology. The nice thing about that is you don't have to know ahead of time what the particular protein is. You can figure it out based on its mass spec, and you don't need binding agents. You don't need antibodies and aptamers. That's another nice aspect to it. They're competing head to head. I think they're still both being used very, very extensively, but I've been surprised at how much more used the mass spec is. For looking at single cell metabolomics, again, it's mass spec, which is the major workhorse here. And there really isn't anything else right now that works anywhere near as well. Mass spec is being used to look at primary metabolites such as amino acids and sugars and organic acids, nucleotides, secondary metabolites such as hormones and neurotransmitters, and then what are referred to as xenobiotics, or things that are either present in the environment that are contaminating our bodies, or drugs that we take, for instance. So looking at drugs, pesticides, pollutants, all those things are part of the metabolomic analysis nowadays. And what you're looking for there is a metabolomic fingerprint. Again, you're taking single cells, you're disrupting them. We don't have the ability to do any molecular barcode here. We have to look at them on an individual basis. So there are a lot of different mass spec runs to look at single cell metabolomics, which of course the mass spec companies are very happy about. And then there's statistical analysis of these different cell types, subtypes within a larger population on a metabolomic basis. At the end of the day, what we're trying to do is put all that back together in terms of the protein state, the mRNA state, genetics, as well as metabolome, and ask ourselves, what then are the characteristics of this particular cell and what are the characteristics of that within the overall context of the tissue from which it is derived. So that's the way spatial biology is going. So I mentioned so far the sort of classic reductionist approach where we take the tissue, we try to isolate the cells from within it, and analyze the cells one by one. But a major transition which is occurring right now is much less reductionist, and that is actually analyzing the cells for their content in situ, that is within the tissue. So this is where companies such as 10x Genomics and NanoString are using RNA analysis, DNA analysis, and protein analysis, all on tissue slices. And what they're able to do is look at hundreds to even thousands of different targets of each molecular type within a cell simultaneously and visualize that, understand the morphology of the cell and how perhaps the biological content is modifying that. All of that then becomes quite interesting because you're looking at the tissue sample itself, all the different cell types within it, and able to visualize the differences and determine in each cell what the particular mRNA that's present, the protein which is present, how much of them, and compare and contrast across that tissue sample. And then compare across different slices of the same tissue and tissue to tissue. So, in my opinion, this is a major advancement that we're just beginning to see the benefits of. And as we begin to understand the spatial biology on the cellular, organ, and total organism level, we'll find new ways and new needs in the diagnostic testing arena. I think the immediate issue that we're going to be faced with, though, is the use of practical samples like blood to access molecular information about what's happening that could impact our health in small subsets of cells in inaccessible sites within our bodies. How are we going to do that? Are we going to be able to find very small amounts of materials from those cells which help to differentiate them? Or, if it's the same thing as other cells and we simply now seeing small changes in a small population of something that's present in that blood sample from other cells, we won't be able to tell what's going on. Or we're going to have to move to something like being able to visualize metabolic changes in small cell populations early enough to intervene with preventative measures. That's a tough thing to do. There are some advances occurring there with ways to label particular types of metabolites and visualize them, but that's not a long laundry list of library of molecules to do that. So I think we have a lot more to do before we're going to be in a position to know overall what the implications are. But I think we in the diagnostic community have to watch what those learnings are from spatial biology and how they will change the way that we develop and use diagnostics. Time will tell, and the changes are coming quickly. What this makes me think about is the allegory of the cave as told by Socrates in Plato's Republic, Book 7. In the allegory, Plato describes through Socrates that there are prisoners who have spent their lives chained in a cave facing a blank wall. They've been there all their lives. They've seen nothing else. They watch shadows projected onto the wall by objects passing in front of a fire that's behind them. They give these shadows names. This is their reality. Socrates speculates that if one of these prisoners had been freed and would look around to see the fire, the light would hurt their eyes and make it difficult for them to see the objects casting the shadows. They wouldn't even recognize what they were. All they know are shadows. If that prisoner were told that what he is seeing is real, instead of the other version of reality that he sees on the wall, he would not believe it. I think, right now, the situation with spatial biology, we are basically being released from the cave. I think that we're able to see objects in a new way, and we haven't figured out how to interpret them yet. That's going to be a long process, but I think it's so important, and I think it's going to lead to a lot of innovation in diagnostics and many other fields as well. And by the way, over time, as we come to grips with what is meant by this new reality, this new spatial biology, we're going to realize that there is yet another cave to be released from that will take us into yet another reality, and we'll have to rethink it again. But that's a lot of fun. I'll look forward to that. Well, thank you once again for watching another episode of Interesting Stories in the History of Diagnostics. This is Mickey Urdea for Halteres Associates. I'll talk to you next time. Bye.